So our last panel for today is going to be on emerging strategies in Duchenne. And so this is going to be a bit of a focus on gene editing and more specifically talking about CRISPR technology. So Ryan and I are going to be joined by Dr. Melissa Spencer from UCLA and Dr. Kevin Flanigan from Nationwide Children's Hospital for a question and answer session in just a little bit. So as we go through this introduction, if you have questions, please submit them to that comment box below. But we are going to begin with an introduction just to kind of help set the stage for everyone, and that is going to be handled by Dr. Spencer. So, Melissa, if you are ready.
Hello, everyone. So today I'm going to give a very brief overview of an exciting technology called CRISPR-Cas9 gene editing. And this technology allows for precise edits or changes in DNA in living cells. Can I have the first slide? So I'm going to introduce you to the basic mechanism of how it works. So I'm representing the two components of CRISPR as a flag and scissors. The flag represents a guide RNA, you'll hear that term, and the scissors represent the Cas9, which is the cutter. It's the enzyme that cuts the DNA. And so they make a complex, and the flag guides the scissors to the precise region of the DNA where it makes a cut. Next slide, please. So the precision in CRISPR really comes from the Watson-Crick base pairing between the guide RNA, which is shown as those blue lines where it says Cas9. So the Cas9 is actually this blobby blue thing in the background, and the guide RNA, which is the flag, is this yellow line with the blue lines up to the top there. So what is being shown here is a guide RNA binding to the DMD gene, making contact in a specific way, and then that Cas9 is going to cut and make a break in the DNA. Next slide, please. And the DNA is repaired using the naturally occurring machinery that's in the cell. And there's a couple ways it can occur. One is NHEJ, or non-homologous end joining, where those two pieces are just brought together. And sometimes you have insertions or deletions, but the platforms can be designed to accommodate that. The other way is homology-directed repair, which requires a third component, and that will completely repair the gene. But unfortunately, this is not a method that's going to be efficient for skeletal muscle. And so most researchers are using NHEJ to develop their platforms for Duchenne. Next slide, please. So how can we design a CRISPR that will work on the DMD gene that would be therapeutic for Duchenne? Well, the theory behind the way most people are approaching CRISPR for Duchenne is similar to what you probably heard about with exon skipping, where in the case of DMD, there's no protein produced. There are deletions that prevent that. And but this approach uses the idea that you can turn a DMD mutation into a BMD mutation through reframing, and that's what exon skipping does. And then ultimately you have a protein that has a small region of the protein deleted, but most of the protein is retained. And we know that BMD is a milder form of muscular dystrophy and can have a range of severities from mild to severe. So most researchers are trying to design their CRISPR platforms to make a very mild BMD, so to turn a DMD with no protein into a mild BMD with a small change in the middle of the gene. Next slide. So this can be accomplished a few different ways. You can have single or multi-exon deletion. Both cases you can use two guides around the exon, and so most researchers are targeting the introns, which are non-coding regions. So where those yellow lightning bolts are, those are the flags. And the little Pac-Man, that's the Cas9, that's the cutter. So on the left side, I'm showing a single exon deletion. And on the right is multi-exon which is beneficial because we can make designer dystrophins. So we are not limited to single exon deletions with CRISPR. We can delete multiple exons, which can encompass many patient mutations and can also be beneficial from a regulatory point of view. And if I don't think I've mentioned it yet, but CRISPR is permanent. So it's acting on the DNA to make a permanent change, as opposed to exon skipping, which is not permanent. Next slide. And one additional way CRISPR is being used was developed by Eric Olson, and he's just targeting the splice site of the exon, similar to the way exon skipping works again, but this is permanent to skip a single exon. And occasionally this can give a full-length dystrophin if it reframes the gene properly. Next slide. So CRISPR is working very well in cells and culture, but to develop a therapy for Duchenne, we need to be able to deliver to the body, all the muscles of the body and the heart. And there's two main ways that it's being done now. People are attempting to develop non-viral methods to use nanoparticles. As of now, this is not an efficient system, and these need to be optimized. Sorry. The other is viral vectors, and the common one that's being used is adeno-associated vector. This is the same vector that's being used for clinical trials to accomplish gene replacement. So these gene therapy trials that are ongoing in the United States. So this is a very efficient vector that exploits the biology of viral vectors, of viruses, I mean, and can enter the muscle very effectively. Next slide. So both CRISPR and gene replacement, gene therapy trials are using AAV, but there are differences between CRISPR and gene replacement. And so I just want to highlight that here. So while CRISPR acts on the endogenous DNA and can be tailor designed to make any dystrophin that's wanted using multiple guides, dystrophin replacement with micro or mini dystrophin is actually adding in new DNA that does not incorporate into the person's DNA. It stays outside of the DNA and in the nucleus. And so the other aspect of this that's different is that with gene replacement, in order to fit dystrophin into the vector, as you know, it has to be modified, and the mini and micro dystrophins retain about 30% of the protein. With CRISPR, much more of the protein can be retained because what you're putting into the AAV is actually the gene editing tools. You're not putting the dystrophin. And it's also beneficial because it's using the normal regulatory sequences that normally drive dystrophin expression. And so those are the main differences. The size of the dystrophin that's created, what's actually put into the AAV vector, and the functionality and flexibility that CRISPR provides to be able to make different designer dystrophins. Next. And I thought this is often a question, how is CRISPR different from exon skipping with antisense oligos? Next slide. So CRISPR acts on the DNA, which is the code. We all have it in our cells, in our chromosomes. Whereas antisense oligoskipping acts on the mRNA, which is like the Xerox copy. So CRISPR should be permanent, should not need to be redelivered every week, like the antisense oligos. And then lastly, last slide. So where we are in the field is, next slide please, is that several scientists in their lab have successfully delivered CRISPR components to mouse models. It's looking very efficient in mouse models. There's been one dog study done by Eric Olson where he showed that it does work. You can use AAV to deliver CRISPR components either by intramuscular injection, which is not what we want to do clinically, or by systemic delivery. And he is seeing very robust levels of dystrophin. And so this is extremely promising, but we do have several obstacles that we need to overcome, and I think we'll discuss them in this session. So back to Eric and Ryan, so we can have our our session.
Thank you, Melissa. Really appreciate that introduction. I think that's going to help set the stage for everyone. So we are going to be joined again today by Dr. Kevin Flanigan. And so we have a handful of topics to discuss, and as well, we're happy to take questions from the community. So if you have any questions, please put them in that box below, and we will try to get to them as we discuss some various topics. So Kevin, I'm going to throw this first question to you. As Melissa, showed in that kind of opening presentation, we see a lot of strategies that are looking towards removing additional exons. And I think, when a lot of families consider CRISPR, the hope is that we're going to have gene correction and that we're going to get back to full-length dystrophin. Could you maybe expand a little bit on why we see a focus on these deletion strategies at this stage?
Sure. Well, the first issue is one that Melissa raised. It's more efficient in skeletal muscle. There's a bias toward this recovery pathway that cells use called non-homologous end joining, where the broken parts can come back together. The cleavage site from the CRISPR, from the Cas9 molecule, allows it to come back together. So that works better for thinking about excision of pieces of DNA than putting pieces back in. The homology-directed repair mechanism, which is the primary way that cells can be sort of directed to put in a donor piece of DNA to completely restore a reading frame, is actually not active in the kind of cells that muscle cells are. It's dividing cells, it's replicating cells. Mitotic cells are the ones that make use of the homology-directed repair pathway. So there's a bias to the non-homologous end joining, or NHEJ pathway. So in some ways, the efficiency of it is probably much greater in skeletal muscle. Now, we should point out that there are potential routes to restoring full-length gene therapy for certain mutations. So, for example, mutations of one or more exon duplications are ones where there are potential strategies to excise those duplicated exons only in a way that you would wind up with full-length protein. And Dr. Olson's lab has demonstrated in certain small subclass of mutations, mutations that affect the splice sites where additional pieces of DNA are inserted, sometimes as pseudoexons inserted into a gene. And in that way, you could restore a full-length gene. But as things stand right now, that's a smaller number of patients that will be amenable to that. And early development, of course, for most things is directed to what will be of benefit to most patients. And these kind of excision approaches are likely to be quite beneficial to the largest number of patients.
Great, thank you. I think that really helps kind of clear that up. We did have a number of questions already about, whether nonsense mutations could be addressed with this or duplications, and so I think that's great to see that that's something that we can maybe expect down the line, but at this stage, kind of going for those after maybe the hotspot regions, maybe what we see kind of early on. And I think we also wanted to talk about some of the challenges that we can expect with CRISPR. And I think maybe a first place to start is just it helps to compare to what we already know, and I think everyone is a little bit more familiar with gene therapy. And so maybe we can talk about, does the packaging of CRISPR, with the guide RNA, the Cas9, is that going to differ than what we see with the current gene therapies with the transgene and the promoter? And so, Melissa, I'm going to direct that one to you.
Okay. So just to reiterate, we're not putting dystrophin into the AAV vector like we are with gene therapy. We're putting in these gene editing components. There are a lot of different Cas proteins that come from different bacteria strains. And the one that was initially used for most gene editing is very large. And so it does fit into AAV, but the guides need to go into a second vector. And so this is a problem because we're seeing a lot of issues with the higher doses of AAV. And so I think people are now thinking about using other Cas9s, other editing material that's smaller, that you can go into a single guide system. So the trend is to probably try and fit it all into one using different Cases that are smaller. So packaging is an issue from the perspective of the size of Cas9, but it doesn't have to be modified like dystrophin does. It would be optimal to have a smaller Cas9. So SA-Cas9 is the one that's smaller and fits. So I think I answered your question.
No, you did. Thank you. And if someone were to use one of those, the larger Cas9, would that have then an impact on how efficient the editing capacity is, because we're kind of splitting the amount we're delivering?
Well, so right now we're trying to figure these things out, but there is a benefit to having a two-component, two AAV system, a dual vector system, in that it's been demonstrated now by at least 3 labs that being able to titrate your one vector to the other vector, so your guide vector to your Cas9 vector, actually is beneficial. So scientists have shown that adding more guide vector really improves the efficiency and how long the therapy will last. So there are some issues that people have encountered in long-term studies in animals, showing that there's a loss of the guide vectors. But by boosting the guide vectors, the dystrophin is more efficient, and then the therapy lasts longer. What I mean by that, because that could be confusing, is that if you're not sustaining your guide vector and you don't have enough editing, if you have inefficient editing, you're not going to hit very many nuclei and edit them. You're going to have very low dystrophin. And if you don't hit a certain level of dystrophin, you are not going to protect those fibers. So it's very critical that we reach a certain threshold of efficiency before we go into patients, since right now AAV is a single dose entity. So a key thing is getting that efficiency up. So the double guide system does seem to help. However, you have a double dose of AAV, and we know that's bad. So the SA-Cas9 will allow lower doses to be used. It's not clear how that's going to impact the guide ratio or the vector stability. That was a bit long-winded, but
no, I think that's very helpful, and I think that was a clear explanation. So, Kevin, then we expect that some of the same immunological challenges that we're facing right now with gene therapy and concerns about pre-existing antibodies, and those are still going to be challenges that we're going to face with CRISPR therapy when we get to in-human trials.
That's right. We do expect some of the same safety issues, although we should say that Cas9 and genome editing presents its own extra set of safety issues as well that have to be addressed. So once we put aside AAV immunity, which may have more to do with efficiency of these approaches than with safety of these approaches, there are issues related to immunity to Cas9, which is these come from bacterial proteins, and we don't quite know yet what really the certain sort of how much Cas9 immunity there will be in the population who we want to treat with these. But the biggest safety issue has to do with when we put in something that we're intentionally damaging DNA with, we have to make sure that it's absolutely specific. So we worry about the off-target effects of this as well. If we potentially, we're intentionally damaging DNA and letting the cell repair it. If we do that by accident at the wrong spot, we could do it at a place with a gene, for example, that suppresses tumors or something like that. And we want to make sure that we don't do that sort of DNA damage. So the major safety issue is really understanding off-target effects from this. And then, as Melissa mentioned, the goal is to have this express and be long-standing, to have Cas9 in cells working for a very long time. So you can imagine as we do this genome editing, genome nicking over a long time, the DNA breakage over a long time, we accumulate those risks over time. So it does present some challenges as we think about sort of safety studies that we have to do on the pathway to getting to clinic with it. So we have some of the same issues related to AAV, to dose, maybe challenges with the fact that we're using 2 vectors, so we are We might have to give twice as much AV in the end to get these effects. We don't know all those answers yet, but then this own set of its own kind of risks for it that we have to be aware of.
And could we maybe just expand on exactly what we mean when we're talking about off-target effects, just so that way, anyone who's hearing about it for the first time is kind of crystal clear on what we mean?
Sure. I'll start, and Melissa, I'm sure we'll jump in. But what we worry about, the component of this, the guide RNA component, is a specific location within the genome, within the chromosome. In fact, we look for very specific places within the DMD gene locus, and that's a sequence of nucleotides like DNA that are complementary. They're RNA, and they're complementary to a location. They pair up with a portion of the DNA that we want it to match to cleave. And by chance, there's some regions of the DNA of our chromosomal DNA that's more specific, more exact, only there at one place in the entire human genome, and that's what we want. But it's not really an all or nothing response, really. So near perfect pairs, for example, over these 20 nucleotides or so, could cause a risk for off-target effects. These are things that have to be studied empirically. You have to do these studies in cells and determine how many off-target spots you have where cleavage might occur. And Melissa probably might expand upon that.
No, perfect.
Okay.
Great. Actually, we had a question come in. Melissa, could you maybe just kind of further expand on what you mean when you say permanent? I think there was just a little bit of uncertainty with that.
Right.
So that would be great.
It's a little bit of a difficult concept. So the permanent idea is you are having cutting of DNA in the nucleus and either NHEJ or HDR. So you're having some editing occur. When I'm saying it may not be permanent, what I mean by that, and I think I probably confused everyone, is that you can lose those edited nuclei if the muscle fiber does not make enough dystrophin. That was my point. And so it's very important that the efficiency is high. So if you are editing nuclei, but then your muscle cell is dying, then those are going to disappear. And that's what I mean. So it is permanent, but we need high enough efficiency to retain the nuclei in the muscle fiber.
With that, do you think the age of the patient is going to factor into where CRISPR is going to be more appropriate with growth and maturation? How will that factor into potentially looking at cohorts that would be most appropriate for a therapy like this?
So ideally, you would want to treat young patients prior to disease. That doesn't mean it won't work for older patients. There are issues with AAV and the immune response that make one want to go into younger patients right now until we can address some of the safety issues. And so I think it could be used for the whole spectrum of disease, but most likely is going to be most effective in the younger patients. And there's another element we haven't discussed, which is targeting the muscle stem cells. If we can target the muscle stem cells, then we can have a long-lasting therapy, because those cells, if they're edited, can divide and make more daughter cells. And so I've lost my train of thought. It's like, yeah, so we would want to target those muscle stem cells. It gets harder in the older patients, but not impossible, because there is a lot of fibrosis. And there was another point, I'll think of it.
Kevin, you had something to add?
May I jump in just with a thought about stem cells in general? And it's just something that I from the clinic that I've heard. Sometimes parents come in the clinic and they say, "Oh, I've heard about a therapy done with CRISPR for another disease." Mm-hmm. And it is worth noting that where things so far have shown success is what we consider ex vivo therapy for example, kinds of blood disorders where cells can be taken out of the body. Yeah. Editing can be done to those cells, kinds of stem cells, in a test tube, and then those can be tested for all the things we've talked about, for off-target effects. We can make sure people can make sure that there's no detrimental effects, and then those patients' own cells, now corrected put back into the body. And we're not at a place, obviously, where we can do this with muscle cells. We can't take out muscle stem cells, correct them, and put them back in the body. And I only add that in after Melissa's thing, just because I know that's sometimes confusing to parents when they read about it being done for a clotting disorder or something else.
Sickle cell. A lot of people were reading about that with excitement.
That's exactly right.
You see a story on the news that this has cured a patient, and it's everything this community wants, but nothing they can have right now. And with that, it's the big question of timing. And I know this is a hard question, but you hear debates over in the next 2 years, we're going to dose a patient with CRISPR, or maybe it's 5. And I know it's difficult for you to sort of, say it's going to be exactly this long until we have trials, but can you talk about this a bit, about the steps that need to be taken, and what are your thoughts about the next couple of years with trials with CRISPR?
We'd love to hear from both of you. So Kevin, we'll have you start, and then we'll kick it over to Melissa.
Yeah. Sure. So, well, the first thing to remember is, although now from a lot of different studies, we as a community or field, we have a good sense of the promise of gene replacement therapy, and we have some sense in learning more, unfortunately, about the toxicities associated with that as well. What we're doing here is fundamentally different. We're trying to go in to edit chromosomal DNA accurately. So it is, although the idea of giving AAV components to people gives us some sense of how the FDA will respond to things, the kind of safety issues we'll look at as investigators, there is a whole another set of safety and efficacy issues we have to really take care of first, we as a community. Now, I'm just reluctant personally to make predictions because awfully good scientists and now awfully good companies are working on this, and everyone is working very hard on it. And the joke is we always overestimate what we're individually going to do in our labs and underestimate what the field is going to do over a period of time. I can promise parents that there is serious work going on in a lot of academic laboratories and now a lot of commercial laboratories as well, and clear pathways to understanding what we need to do for this. In the meantime, what gene replacement therapies have taught us is what are the kind of proper outcome measures we should be using in trials for patients. We have a good sense of what the relative safety is from the AAV vectors itself, and not a perfect sense, but really an opening understanding of what those are. So I do think we're on the scale of years rather than decades to one of these trials. I think that's about as much as I'm safely willing to predict myself. I'll hear what Melissa. I look forward to what Melissa has to say.
I don't have too much to add, except I think we'll know more as things proceed in other diseases. So for these blood-borne diseases where cells are taken out, edited, and put back in, the regulatory pathway will become more clear, and that's going to lower the threshold. So every one of these successful trials then makes it more likely that we can speed up. And it's really one of these situations, having been in the field so long, we just never know when it's going to happen. We thought gene therapy was happening 15 years ago. It didn't happen right away, but then all of a sudden the path was cleared from the SMA studies for systemic gene therapy, and now everybody is excited and things are moving forward. So these things tend to sort of come in fits and starts. You think nothing's happening, but scientists are working very hard in their labs trying to get through some of the regulatory issues, some of the safety issues, optimizing their therapies. Sometimes they fail, but it's always, one step back, 2 steps forward. And it's really hard to know. But I think we'll know more as these other diseases start to apply CRISPR.
Okay, great, thank you. We did have a lot of additional questions, and I think this is something that's definitely going to warrant some follow-up, and I think maybe we can look towards doing an additional webinar with both of you, because this was some really great content. I do have one quick question to throw to Kevin. Something we hear a lot from families, and I think is a really important issue, and it's something we see in our preference survey work as well, is about waiting for CRISPR, and just wondering what kind of your conversations look like when you speak with families in the clinic that are maybe kind of waiting.
Right. Well, it's a great question, and I often, when I talk about CRISPR, I remember when the New York Times had a article on their science pages about 3 CRISPR papers in Science, and about 3 years ago, and people came into the lab the next week with that paper in hand and said, or into the clinic and said, okay, when can I get CRISPR? I think it's important for parents to really understand. I think it's important for us as clinicians and scientists to educate the community about what's different about it and the challenges that exist for it. I think that I often have try to have conversations in clinic to lay out what we've learned, what the newest things are, and the differences between correcting it in a mouse or a dog versus what that means for scaling up to humans. That's really quite important. There are also, I think, ethical and societal issues that people are concerned about with CRISPR. It won't keep us from finding a way forward with it. But when we start editing the genome, we worry about what we're doing, what editing we pass on to the children. What do we edit the germline as well, or just things in the target tissue like muscle? That's a big topic and beyond the scope of this question itself. But there are some great resources, I will say, for parents out there. I often refer parents to what I think is a wonderful book by Jennifer Doudna, Doudna, who's one of the scientists really responsible for clarifying CRISPR mechanisms in mammalian cells. And she wrote a book called A Crack in Creation. I recommend it. I don't get kickback for recommending it. I mean, it's out there on Amazon and paperback, but I think it's for the interesting parent, I think it's a really good description of what this really remarkable technology is. It's become, a great tool for laboratory research and our great hope for clinical research as well. So I would recommend that to anybody.
Great, thank you. And thank you both for taking the time to talk about such an important topic with us and with the community. We really appreciate it.